lab space isn't exactly what you would expect in a typical lab. I'm sure you guys all have visions in your head. Everything's white, everything's sterile, everyone's wearing lab coats and scurrying around. But here it's a little bit more open. And the reason that we keep the labs open is to try and get people to talk to each other. Rather than run into your office, do all your work, and emerge 8 hours later blinking into the sunlight, then you want to have people interacting, having people get to know each other, ask questions. What are you working on? What didn't work? Do you have a chemical I can borrow? What's your expertise, and how can I use it? So in this lab, this is Kirk McManus's lab, and he's looking for ways of detecting colorectal cancer at the genetic level, looking at the individual changes in the genes, if there's multiple copies of a gene, or if there are two genes next to each other, that if they weren't next to each other, you wouldn't have cancer. But when they're side by side and they're expressing something that interacts, that causes the changes in a cell that can become sort of the tipping point to carcinogenesis. So this is Kirk McManus's lab. The next lab over is looking at blood disorders. And the one down at the end of the hall is leukemia. So they're all cancer, but they're all different. But the students and the trainees from here can walk over to the other one and say, well, this isn't working for me. When I did my Western blot, this happened. Can you help me? And so this way we can start sort of mashing ideas together and getting a better idea of, what changes happen in colorectal cancer. Well, maybe that works too in breast cancer. And those guys are upstairs. I'll go talk to them. Right? So if we didn't have an open lab, people would just sort of shelter themselves off. But we have these open areas, and it gives an idea, it gives us a chance to talk to each other. So we'll come down this way. Normally this place is buzzing with activity, and I mean it's really loud in here. At any point in time we have over 100 trainees. In the summertime we can have another 100. So throughout the year we can have over 200 trainees from around the world, starting from high school students. We try to hire the rock stars of the science geeks in grade 11 and grade 12 and get them into the lab and get them enthusiastic about science, and especially about research. And then we go all the way up to the postdocs. These are people who have become career scientists. They've done the best that they can from grade 12 to their bachelor's to the PhD, and now they've gone into what they call postdocing. So they've got their PhD and they still need more training. So they come here because we have facilities and expertise that doesn't exist anywhere else in the world. And I know that sounds like I'm delusions of grandeur, but we really do have some of the leading scientists here. Upstairs, Dr. Sabina Mai and her Genomics Institute has North America's most powerful microscope. It's the first one of its kind to be installed outside of Germany, and it was developed in Germany, so we have the second one in the world. People are now coming here to use it because it is so unique and so powerful that we have access to it, and we sort of take it for granted that we have this microscope. We have the 3D sim, but nobody else does. And it's kind of a point of pride that we have an entire institute upstairs, the Genomics Institute, devoted to looking at the microscopic details. We're looking at the little tiny bits of protein at the end of DNA that can give us an idea of how fast or how aggressive cancer is going to spread. And that's just Sabina's. We also have the mouse functional genome group. We have a breast cancer genome group. And then here we have the leukemia group. Spencer Gibson's specialty is on chronic lymphocytic leukemia. And he works with the clinicians downstairs on the main floor where the blood samples are taken. He gets access to the leukemia patient's blood through our clinical trials group. And he can analyze that blood looking for differences in the proteins expressed on the outside of the white blood cells. So what we found here is that on the outside of a white blood cell, if it expresses ZAP70, kind of a funny name, but if it has more than 20% of its cells express ZAP70, we know that ZAP70 positive people respond poorly to the typical drugs. And by poorly, I mean 6 years difference in life expectancy. So when Spencer can get the blood samples from the clinicians downstairs, we can work with them and tell them this person has a ZAP70 score of 10, give them that drug. If we can tell them it's a ZAP70 score of 35, we have something else to do. And this is what we call translational research, because we're all in the same building and we're interacting with the doctors that are treating the patients and we're getting access to the patient samples. We can make discoveries in the lab, in our wet labs, and then take them right downstairs and actually have a direct and meaningful impact on patient care. So we'll take you upstairs to the tumor bank, which is a lot more exciting than looking at an empty lab.